Hey everyone, this is Dr. Jyoti Bala. I welcome you all on my YouTube channel. And the session we will be discussing about a recent blood test which can early detect toxic proteins years before Alzheimer's disease symptoms. Recently, researcher from University of Washington has published this work in PNAs. As we know, Alzheimer's is a challenging progressive neurological disorder which has limitation of early diagnosis, and patients receive diagnosis of Alzheimer's diseases only after the exhibition of symptoms, which limits the treatment option due to the late diagnosis of disease and further progression of symptoms. There are few factors which make Alzheimer's disease a challenging sector because there is a lack of effective treatment. Alzheimer's disease is also difficult to diagnose, especially in the early stages. The current available tests which are in the market are very costly. The frequent accurate tests which are available for Alzheimer's diagnosis are spinal tap, PET scan, tests to identify the biological changes associated with Alzheimer's diseases, and the biomarkers of Alzheimer's diseases like taut angles. The spinal tap are invasive method, whereas the PET scan is costly for patients, and most of these tests are not covered in the medical care and medical insurance system. Tests are really costly, it's around $1,000. Because of these limitations, either misdiagnosis and late diagnosis happen with the patients. There are only few efficient tests which are currently available in the market, and the top companies which are currently working on Alzheimer diagnostics and kits are C2N Diagnostic, Quest Diagnostic, Rossi and all. According to the Alzheimer Association, nearly 13 million people in USA are expected to be living with Alzheimer's diseases by 2050, and globally the projection is 153 million. This means the early diagnostic of the patients are really very important. In a previous videos, we have already discussed one similar studies where the risk factor of Alzheimer's diseases has been detected via saliva-based kit. I request you to watch that videos also if you are working on neurological projects, especially the Alzheimer's disease starts long before the cognitive impairment symptoms surfaces. The amyloid beta protein that misfold and clump together form an aggregate called oligomer. And these toxic oligomer aggregates of beta amyloids are actually responsible for developing Alzheimer's disease. Hence, the scientists from University of Washington who have published their data in PNAs related to this test. In this lab test, they have detected the amyloid beta oligomer in the blood sample. The test is known as SOBA, which stands for Soluble Oligomer Binding Assay. Briefly, SOBA exploits a unique property of toxic oligomer. It is known that misfolded amyloid beta protein begin to clump into oligomer. They form a structure known as alpha sheet. These alpha sheets are not ordinarily found in the nature, and they tend to bind to other alpha sheets. SOBA is a synthetic alpha sheet designed by this research team that can bind to the oligomer in the samples of cerebrospinal fluid or in the blood sample. The test then uses the standard methods to confirm that the oligomer attached to these test surfaces are made up of amyloid beta protein. This SOBA blood test could aid in identifying the individual at the risk or at the incubating stages of this disease. SOBA blood test has potential in the early diagnosis before the cognitive impairment symptoms and also in the treatment plan of Alzheimer's disease. And the studies, the research team has used this blood test on the 310 research subjects, which include the person with no sign of cognitive impairment, with mild cognitive impairment, and also the samples from the Alzheimer's diseases and another form of dementia. They found that SOBA test detected oligomers in the blood samples of individuals with mild cognitive impairment to the moderate and severe Alzheimer's diseases. 53 research subjects who were diagnosed with Alzheimer's diseases after death by autopsy, and out of this 52 research subjects blood sample which had been taken years before their death also contained the toxic oligomer in the SOBA test. And the blood sample from the individual in the control group who remained unimpaired lacked the toxic oligomer. Now the research team of Daggett University of Washington is collaborating with the university's spin-out company LPAP. Now the research team are trying to convert this SOBA research test into the diagnostic kit for the commercialized purpose. Additionally, the research team also showed that SOBA test easily could be modified to detect the toxic oligomers of another type of proteins associated with Parkinson's disease and other dementia. because the accumulation of toxic oligomer that forms alpha sheet structure are not only associated with Alzheimer's diseases, but they are also associated with Parkinson disease and type 2 diabetes. SOBA test has potential scope in the disease related to protein misfolding. I hope you have liked this session. Don't forget to like and subscribe the channel and do share these information among your scientific endeavor. Thank you so much.